Hi, my name is Jeff Groh from Calmetrix, and I want to welcome you to the webinar on the study of food safety and spoilage by isothermal calorimetry. There are 5 things I want to accomplish today. First, I want to ground you basically in what isothermal calorimetry is and how we can apply it to the study of food safety and spoilage. We'll talk a little bit about the instrument itself and why you might want to add one of these devices to your laboratory. And then I'll finish up with some tools for additional learning. This is a schematic of an isothermal calorimeter showing the primary components. Let's start on the inside of the instrument with the sample, which goes inside of a 125 milliliter sample cup. And I'll show you pictures of the sample cups here in a little bit. We typically run this instrument in a differential mode, so we're running a sample against an inert metal reference in a differential fashion. Both the sample and the reference are sitting on top of heat flow sensors, which are in turn connected to a large metal heat sink. This basically then constitutes the actual calorimeter, which is sitting inside of a temperature controlled chamber shown in blue here. And we can control the temperature in that chamber very precisely from 5 to 70 degrees C. So typically what we're doing is running this experiment in an isothermal mode. The sample tends to be pretty massive, 125 milliliters with sample with the types of materials we're dealing with. You know, it may be in the order of tens of grams to hundreds of grams of sample. So the sample is pretty massive, and the actual calorimeter is massive. So temperature equilibrates slowly in these kind of devices. So we typically run in an isothermal experiment or an isothermal mode and look at the data as a function of time. So let's look at the sample and what might be happening in the sample. There could be 2 things going on. One, the sample might be giving off heat in some way. For instance, maybe microbial activity or other life processes that are going on that are giving off heat. So those are exothermic events. Or there might be endothermic. The sample might be absorbing heat. So in the case of exothermic, as that heat is evolved from the sample and the temperature goes up in the sample, that heat flow flows from the sample through the sensor into the heat sink. So there's a heat flow from the sample into the heat sink. The heat flow sensors, which are basically thermoelectric devices, measure that heat flow. The raw signal coming off these sensors is a voltage signal. And through the manufacturing process, when we calibrate the instrument, we relate that voltage to a heat flow in watts. So typically the data we're looking at then is watts. We, in most cases, will normalize that per gram of sample. So we normally present it as watts per gram, power in watts per gram, as a function of time. And as you can see here, the time can be quite long. So we run experiments anywhere from 24, 48, 72 hours and longer. Sometimes we run experiments for days, up to 20, 30, 40 days in some cases. It's really no limit from the instrument standpoint. It's really more application driven in terms of the signal that you're generating and whether you've got enough sensitivity to look at the data. So we use the data in a couple different fashions, a couple different ways. We might qualitatively use the data to look at a good versus bad. So there we're just looking for the sample shape or the curve shape. Other times we might be wanting to quantitatively compare samples. So we might be looking at quantitative differences in when things happen, the time to various peaks in the curve, different types of events that are happening. So it's both a qualitative tool and a quantitative tool. I've blown up the beginning portion of this curve, which illustrates the point that when you drop a sample into the calorimeter, the sample is never at the same temperature of the calorimeter, so there is a time associated with the sample coming into equilibrium with the environment in the calorimeter. And that might take a couple hours, depending on the mass of the sample and the heat capacity and so forth. So you just have to bear that in mind from a experimental design standpoint and from a data interpretation standpoint, and realize that these startup events are going to happen at the beginning of the experiment. Now there are other things that we do with the data. For instance, if you integrate power over time, you get energy. And so another type of data that we look at are these continuous integral curves. So here we're plotting energy in joules per gram as a function of time. And you see smoothly increasing curves here. That's because these are continuous integrals of these curves. So if you go to 10 hours, here it's basically taking this curve 10 hours and integrating under this slice of the curve and generating that as a data point at 10 hours. So that's what these continuous integral curves are, and we use them in the same fashion, either from a qualitative standpoint or quantitatively. We might go to a particular time and look at the total heat evolved at that point as a way to compare two samples at a particular time. It's a good spot here to point out that this technique is considered a non-specific method. And what does that mean? It means that the calorimeter doesn't care where the heat's coming from. All it knows is that the sample is generating heat. But if you've got 2 or 3 or more processes going on, it can't necessarily differentiate. So it can tell that there's heat coming off the sample, but it can't necessarily say that the reason that there's this bump in the curve is because of a specific bacteria or whatever. So you have to sometimes, based on the experimental design, you have to use good experimental design and test matrices in order to pull these things apart, or use the instrument in combination with other techniques. So it's very important to remember that this is a non-specific technique. A question that always comes up is, well, I have a calorimeter in my lab. How is it different from what I've got? On the left is the typical data that you get from an isothermal calorimeter. So here we're looking at heat flow as a function of long time frames. And over on the right is typical data from another very common calorimeter that you might be familiar with, and that's a differential scanning calorimeter. These instruments are both the same in the sense that they both measure heat flow as a function of time or temperature, but that's where the comparison stops. They're really used for totally different applications. In the case of an isothermal calorimeter, we're using very large sample sizes in the order of tens of grams, and we need large sample sizes because we're typically dealing with inhomogeneous materials, and secondly, the reactions that we're looking at are very low amounts of heat per gram. So by using very large sample sizes, we can get a high absolute signal that then the instrument can pick up. The experiments are very long term. They're long isothermal experiments in the order of tens to hundreds of hours. Again, because the things that we're looking at here happen over long periods of time. They're not short-term events. So we need long isothermal experiments, and the instrument is designed for that. And then we're also usually interested in a fairly limited temperature range with these applications, say from 5 to 70 degrees C. As opposed to DSCs, which use very small samples, typically in the order of 1 to 20 milligrams, so you're talking an order or two of difference in sample mass. They're very short experiments because the mass of the sample is small and the system is small. We ramp temperature in these devices typically. They can change temperature very rapidly. They can run isothermally, but normally they're running in a temperature ramp mode, say 10 to 50 degrees C per minute, so the time frames are very short, and the temperature range is usually pretty broad from minus 150 to greater than 700 degrees C. These instruments are primarily used for looking at changes in heat capacity in samples as a function of temperature. A lot of applications in the polymer industry where you're looking at, for instance, glass transition or other types of crystallization and melting phenomena in polymers. Totally different applications. You would never use these devices for a large, for an inhomogeneous food material. You just wouldn't have the sensitivity, and vice versa. You wouldn't use an isothermal calorimeter for trying to do a heat capacity or a glass transition on a polymer. So these systems are designed for what we're going to be talking about today. Like other analytical instruments, baseline is very important. The isothermal calorimeter is designed for very long-term baseline stability, and that's very important. It basically provides us the sensitivity to look at very small changes in heat flow over very long time scales. So, of course, there's two aspects of sensitivity. The raw baseline noise is one issue, but just as importantly is the long-term baseline stability, because these reactions that we're looking at are very small changes in the baseline, and so a flat baseline is important to pick up, to have the sensitivity that we need for these small changes over long time scales. Sample preparation is very easy. We use 125 milliliter sample cups, so you can get a feel here with this ruler, the size of these cups. It's very easy to get samples in there. It can accommodate large samples, especially in the food industry with the types of materials we're dealing with. It's very easy to get the sample into the cup. They're available in the stainless steel with a Delrin lid, which can be autoclaved. They're also available as a plastic snap lid, which are easier. Both types are reusable. The main issue with the cups is understanding what you want to do from a sample environment, whether you want a closed environment or an open environment. This cup here, the stainless steel with the O-ring snap lid, normally does not have a hole in it. This one's shown with a hole in it, but without a hole, when you put this lid on top, it's a sealed up container. Otherwise we can put holes into the lid, we can plumb in different types of gases. So it really comes down to understanding what you want to do as far as the sample environment, whether you want it in a closed system or whether you want it in an open system, and in an open system what kind of gas you want around the sample. So how does all this relate to food safety and spoilage? Well, the bottom line is that all microorganisms generate heat, and that's what we measure with the calorimeter. So it's pretty simple to understand in the sense that everything we're doing here is generating heat, and that's what the calorimeter is sensitive to. So how might we use this in food science? Well, it kind of breaks down into two main categories. One is this area of food safety and spoilage, which is mostly what we're talking about today. Looking at microbiological activity, bacteria, fungi, yeast, and the heat signatures related to those microbial species. Of course, much of what we do in food science is really how to preserve against activity, microbial activity. So measuring the effect of various preservation techniques, estimating shelf life, and then finally, there are great applications for measuring metabolic responses in fruits and vegetables and correlating that to perishability. So, that's another big area in terms of food safety and spoilage. Another area really kind of is associated with process efficiency and cost. This is where you get into, say, fermentation studies and optimizing fermentation. Seed germination kind of falls into this category, though there's a lot of overlap, because if you've got surface contamination, surface disease, then that's really microbiological activity. But the life processes associated with seed germination normally is a process type issue in terms of, efficiency of germination and so forth, and studying germination. Yeast studies, optimizing dosages of preservatives from a cost standpoint. So these kind of all fall in the process efficiency cost. There's a lot of crossover, interrelationship between these two applications, but in general they fall into these two categories, and we're going to mainly be concerned today with food safety and spoilage. And really, when you get into food safety and spoilage, it's all about preservation primarily. How do you preserve samples? And there are a lot of preservation techniques that we use, a lot that are being investigated. Of course, the big area now is investigating more natural preservatives rather than chemical preservatives. So a big area of what we do in the instrument is in the food preservation area. So how do we look at that today? Well, there are a couple different ways. We obviously use a lot of microscopy techniques. There are a lot of downsides to this technique. It requires a transparent substrate. It is destructive. You're only looking at the surface typically. You're not looking at the bulk of the sample. The techniques can be very labor intensive. It takes a lot of training and a lot of labor to do the tests. They're in many cases not continuous measurements. They're more discrete measurements, which also relates to the labor in the sense that you might start an experiment on Friday, but you have to have technicians or students come into the lab on the weekends in order to collect data points, and then you might miss something between these discrete points. So there are a lot of disadvantages. Similarly with, we do a lot of cell culturing. Folks do that. to investigate these things. Again, that can be a cumbersome technique, requires a lot of prep work, a lot of training. Not all bacteria can be cultured. Again, very labor intensive, takes a lot of effort, and again, in many cases, not a continuous measurement. So there are a lot of downsides to these traditional techniques, and that's really where some of the advantages for the calorimeter come into play. The calorimeter is very easy to use, so much easier to train folks to use it. It's very general in the sense that all microorganisms produce heat. It's a bulk measurement, so you're looking at what's happening in the entire sample. It's continuous, so once you start the experiment, you start it on a Friday, you're collecting all the data, you come back to the lab on Monday, so it's continuous. Not only do you reduce your labor, but you've got all the data and you're not missing anything. And it can save a lot of time, related to this continuous experiment feature. You are not requiring as much labor, but above and beyond that, you can run parallel experiments, so you can collect a lot of data simultaneously, which saves a lot of time and improves, the ability of expanding your test matrix. So a lot of advantages to the calorimeter. I would always position the calorimeter as not necessarily a replacement for everything that you're doing today, but it's a great addition to your lab, an additional tool that you can use in concert with your other techniques to probe what's happening with your samples. One question that comes up in many cases is, well, how many bacteria does it take to get a signal? Really what we're talking about is the limit of detection, which is the minimum change in the measured signal that you can differentiate from the noise over some period of time. In our instruments, we have a limited detection of about 5 microwatts in a well temperature controlled lab. So in a typical microbiological lab, where you have good temperature control in the order of plus or minus a tenth of a degree C, then you're going to get this kind of level of limited detection. It really has to do with the baseline noise, and so our limit is about 5 microwatts. So how does this relate to the number of microorganisms that you can detect? Well, different microorganisms, first of all, release different amounts of heat, but if you use a good approximation for a bacteria that it releases about 2 picowatts of heat, that's the first point. Secondly, in terms of looking at the minimum detectable number of bacteria, you also have to consider the sample size. So in our calorimeter, we've got 125 milliliter sample vial. So if you consider that you can use, say, a 100 milliliter sample, and you work through the math in terms of the 5 microwatt limit of detection and the 2 picowatts per bacteria and 100 milliliters, that means we can detect down to about 2.5 times 10 to the 4th CFU per milliliter. And that that compares very well with other techniques. For instance, it's a couple orders of magnitude better than what you might detect from, say, measuring turbidity. So it is a very sensitive technique. In many cases, you can sense the heat coming from the activity well before you can sense other parameters that you might measure. So if you look at the value proposition for calorimetry in this area of spoilage and food safety, you've got the fact that it's continuous measurement, very minimum effort, easy to use, and easy to interpret the data. And what do I mean by that? Well, if you look at a typical curve here from the calorimeter, this happens to be a juice, probably like a cranberry juice, something that's very resistant to microbial activity, has a high pH, and then compared to a sample that's been contaminated. And you can see the activity, the microbial growth in the contaminated soil. And if you compare that to what you might classically see in your data, it looks very similar, especially when you start thinking about the log, the exponential phase of the curve where things are growing. These curves are shaped very similar. It's just that rather than looking on the y-axis at the number of cells and the doubling of cells, we're looking at thermal power and the fact that a doubling of thermal power relates to the doubling of the number of cells. And the point is that you can use all the same equations that you use right now for studying your curves. And you can infer the doubling times and by looking at these exponential portions of the curve and use all the same equations that you're used to using. And here's an example of that. This happens to be on watermelon pulp that we're working in concert with NC State on, and here two different samples. We're going to focus here on the initial part of the curve, and that's blown up over here. And so you can see here, if we look at the doubling of the population and we go to the curve and we extrapolate down, we can tell that in both cases, we had a doubling time of about 2 hours, but there was a lag between one sample and another of about 10 hours. So there was a different initial population. And how might we quantify that? Well, we can use the same equations that you're used to using. Here we're using a very simple equation. You can use whatever equations that you're comfortable with, but in this case where the A is the initial microbial population and K is the time constant, and then calculating through, the doubling time is defined by this equation, and then we can go to the graph and infer the doubling time of about 2 hours, solve for K, work through the equation, and determine that there's about a 32x difference in the initial population between those two curves. So the point of this whole argument is that you can use the data just like you use your existing data, use the same equations to treat the data, and infer the doubling times and populations and so on and so forth. So how might we use this, in applications? Well, obviously it can be used in a qualitative fashion. This is an example in carrot juice. The green is the carrot juice right out of the container. This happens to be an organic carrot juice. I did not take care with the sample cup to decontaminate it, so there could have been some, contamination in the cup itself. But you see activity here in the carrot juice. Carrot juice is a very verdant environment for microbial activity compared to a sample that was purposely contaminated. So it's very graphic in terms of being able to tell the original sample from a contaminated sample. And so we use the instrument in a lot of cases in a QC way, just to tell the difference between a quote-unquote a good versus a bad sample. But of course, in a lot of what we do, we're interested in how do we preserve materials and reduce this microbial activity. And so the whole concept of using the instrument for studying different preservatives is a big area, and that's what this data is meant to illustrate. There's a lot of data here, so let me walk you through the argument here, the progression. First is the plain juice. This happened to be a melon juice, which was chosen because it basically, like carrot juice, is a very good environment for microbial growth. So it was sterilized in some way and run, and you can see actually it's underneath this green curve. You can't really see it, so there's really not a lot of activity. There's no real power curve here. It's pretty, at least over this time scale, pretty stable. So that's the plain juice. The next curve, the blue curve, is the juice plus a contaminant. In this case, it was a very specific cocktail of contaminants that included some bacteria and some fungi and some other things that they were concerned at in terms of looking at. So that's the blue curve. So you can clearly see the activity, the microbial activity in this blue curve. And then in the yellow and the red curve show the individual addition of two different specific types of natural products that were being studied for as preservatives. One happened to be an extract from the shrimp processing industry, and the other was an extract from a thyme plant, basically. So these were two different natural-based preservatives that they were studying, and that's what the yellow and the red curve is. But you'll note of interest here is that when they were added individually, the actual microbial activity was greater than with the contaminant sample alone. And presumably what was happening here is that in isolation, these materials were affecting one portion of this contaminant, but allowing other things to happen to a greater extent. So actually they made things worse, but when they were actually combined, and that's the green curve, when the two were combined together, then they actually were very good as a preservative. So this is a graphic example of how you might use the instrument to study different types of preservatives and the interaction between different types of preservatives in very complex situations. So a good example of how the instrument might be used to study preservatives. This is an example in the meat industry. This is spoilage and ground beef. So the green curve is plain ground beef, and you can see the spoilage occurring here. This is a blow up down here, and the point here is that, you know, with this technique, you can see activity well before you might be able to see things like color changes and so forth. So it's a very sensitive technique. So that's the green curve is the plain ground beef, and then in the yellow curve, that same material was wrapped simply in cellophane. So it just illustrates how packaging can be studied. And really you have great flexibility because of the size of the sample container and how you want to try to approach those studies in terms of studying different types of packaging materials, barrier materials, and so forth on you know, the protecting food products from spoilage. So a great example of how the instrument might be used in that context. And kind of the final application I want to go over is this is using the instrument for plants and vegetables, harvested plants and vegetables. And of course, in plant, you have to understand that there's photosynthesis, respiration, and transpiration processes going on. But the fact is, in fruits and vegetables that are harvested, the respiration process continues. And the first point here is that you have to understand that if you're running in a closed sample container, you have enough oxygen in there to yield about 400 joules of energy when it's consumed. So the fact that you do have respiration, you have to realize, are you running in a closed container or are you running in an open container, and what does that mean? So in a closed container, you have enough oxygen to consume about 400 or yield about 400 joules of energy. And secondly, there's differences in fruits and vegetables that you have to be aware of. There are fruits and vegetables that continue to ripen after harvest and have an elevated respiration with ethylene production. So here's some examples of those kind of fruits and vegetables. And then you have others that do not ripen after harvest, and actually the respiration gradually declines. And here's some examples of those materials. So when you're running fruits and vegetables, it's very important to understand this concept of respiration and the differences between the types of fruits and vegetables you might be working with. So here's an example of that. This happens to be in an example using blackberries. Again, there's a lot of data here, so let me walk you through the progression. The first run here are plain blackberries that were run in a stainless steel cup with a hole in the top of that cup. So it was basically open to the environment, so there was plenty of air getting into the sample. And you can see, you know, there's exothermic activity, and it continues to advance as time goes on. So, you know, normally blackberries, the respiration would fall off as a function of time, but the fact that heat flows continuing to go up, there's other things going on. And in fact, when you look at the sample after the run, there was obviously mold growing, so there were other things that were happening in the sample. The second sample here is the same berries in that that open cup, but now the berries were treated with lemon juice, and you can see the change between the plain berries and, you know, and the berries with the lemon juice. It definitely affected, particularly the mold growth when you compared the samples at the end of the experiment, and affected the total amount of energy that was being given off. Then the next set of experiments were done in a closed container. So the green curve is the plain blackberries in a closed container. You can see how the respiration falls off here. It's interesting to note that if you integrate under this curve, you'll see that this relates to the 400 joules of energy that the oxygen was consumed and in the respiration process. And then all the oxygen was used up and the activity fell off. In the yellow curve, it was treated with lemon juice. Again, the total area under the curve is still related to the 400 joules of energy. However, there were differences in the two curves in terms of that process, that respiration process. So the conclusions on a limited basis here, based on this limited amount of data, the exothermic energy that we see in the green and yellow curves is associated with the consumption of that oxygen in the closed containers. Number 2, in the red and the blue, the lemon juice impacts the respiration process. Well, in both cases, actually, it affects the respiration process by some mechanism. We don't really know what it is. It reduces it in both cases. And then the lemon juice hinders the mold growth, as illustrated in the red and blue curves. So, great example. Lots of people have done things with the calorimeter on fruits and vegetables, and this just illustrates the types of things that you can do and with fruits and vegetables. So hopefully through these various applications I've given you a sense how great of a tool calorimetry is to assess a lot of things in food science in terms of looking at microbial activity in foods, studying different types of preservation techniques, assessing shelf life, looking at packaging effectiveness, and studying the spoilage and respiration in fruits and vegetables. So a lot of flexibility. I will point out here one of the great things about this technique is it is very flexible and really only limited by your imagination in terms of the types of things that you can do with it. You've got a very large sample container. You can do a lot of creative things in terms of how you expose the sample to different types of environments. So it's just a fabulous tool for you know, a lot of areas within the food science area. And as I mentioned at the outset, you know, it's a great tool in combination with some of the other classical techniques that you've always used. It's not a panacea; it won't solve every problem, but it's really a great addition to your toolkit, so to speak. All right, let's talk a little bit about the instrument. There are two types: the 2000 and the 4000. And this is all the list of the types of applications that we can do. Of course, it's, you know, just a partial list. It really, again, is very dependent on your imagination. But these are the ones that kind of come up over and over and over again. And basically, both instruments can do any of the applications. There are differences. The 2000 has 2 channels, so to speak, so you can run 2 samples simultaneously versus the 4-channel device can run 4 samples simultaneously. So there is a difference. You know, this is double the throughput basically. And there are other differences. The 2000 is considered our highest performance calorimeter. It does use 2 of the 125 milliliter sample cells. The sample cups themselves between the 2 instruments are identical, so that's the same. This uses 2 of them simultaneously, so you can run 2 samples. Temperature range 5 to 70 degrees C. This is actually the same between both instruments. And the duration of test anywhere from hours to days. Again, only limited by the tests that you're doing. It's not limited as far as the instrument is concerned. And that's the same between both instruments. But the 2000 definitely has the best baseline performance, so it has the maximum sensitivity. So if you're looking for the absolute maximum sensitivity, then the 2000 is the best instrument. It's optimized. It has 2 individual calorimeters, so it optimizes for crosstalk, and that's why it has the best performance. It includes, as does the 4000, the software and a computer. So these are turnkey systems. The software is pre-configured, pre-installed. You get the system, you can install it within 30 minutes basically. Get it out of the box, set it up, and you're off and running. So it's very easy to install this device. 4000 again is our high performance instrument. Uses the same type of sample cups, but it has 4 samples, so you can run 4 samples simultaneously. So it's got double the throughput, same temperature range, same duration of test. Excellent baseline for sensitivity. Really, in 99% of the cases, it's probably going to be fine as far as baseline performance. You know, really is context specific on your application in terms of whether you need the 2000 or the 4000 is sufficient. Of course, the 4000 can run 4 samples simultaneously. It includes the software, as does the 2000, and a computer. So again, it's a turnkey type of system. Software is included. Very straightforward software application. It's a single application that sits on the desktop. It has a reports function, which is where you open up up to 8 data files, overlay data, do the data analysis, and also a logger tab which is basically the instrument control. So this is where you set up and run experiments. You can be running experiments and simultaneously analyzing data from previous experiments. So hopefully I've answered this question: why add an isothermal calorimeter to your laboratory? Well, it's a very proven, powerful technique, many good applications, very easy to use. The instrument is something that I can train anybody on, especially compared to a lot of other of the techniques that you're familiar with. It's a very easy to use technique. It's broadly applicable. Really everything that you're involved in is producing heat in one way or another, so it's applicable to anything that's producing heat. So really has broad applicability and really only limited by your imagination. It's very robust. Compared to a lot of other analytical techniques, I have no qualms putting this into, for instance, an academic lab where there are a lot of students coming in and out of the lab. It's a very robust device, but perfectly suited for R&D as well. And it's very cost effective. So for the amount of money that you spend on these instruments, really it's got high value in use. You can get a lot of value out of it for the money that you spend. So it's a very cost effective technique. Perfectly applicable to both R&D and QC. The robustness allows it to be used in QC applications, but the performance is perfectly suitable for R&D type applications. So it's well suited in either application. Can save a lot of time and money while expanding the amount of data that you can get and expanding the size of the test matrices that might be available to you. You don't have labor coming in on the weekend to collect discrete data points, so it saves you money that way and time. You can run multiple parallel experiments, so you can collect a lot of data simultaneously. So big advantages in terms of saving time and money. And it also, in many ways, can reduce your laboratory capital costs because you might not have to add as much other expensive equipment. This is very cost effective compared to a lot of the other microscopy techniques, and especially considering the fact that you can run multiple experiments. It takes up very little bench space, so you don't need a lot of bench space, which affects capital costs. So many reasons why it can reduce capital cost. And finally, I would say that one of the big advantages for our company is our staff in terms of the expertise that we can bring to bear to help customers get the highest value out of the instrument. We have some staff that really were involved with inventing this technology more than 30 years ago. They have a wealth of information in terms of not only calorimetry but all the applications, and so that helps us provide a focus on really providing outstanding customer support. So anybody can sell you an instrument. Really the difference is with the folks that we have available to help you with application support to get the value out of the instrument over the long term. That's where the value comes in. It's not just getting the instrument, buying the instrument, it's the value that you get out of it over time, and that's where our resources can help. Okay, I'll finish up with just some additional tools for learning. You can visit our website. We have great videos on that website that help you understand what really it means to run a sample in these instruments. There are a lot of application articles that you can download. I also can provide you many other application articles that are very specific to your application, so we can help you locate good application material for your specific application. So website is a good source of information. We provide on-site seminars. These are usually done in context after the sale, but they could be done before sale. These are fee-based seminars where we can bring in calorimetry experts and do 1 or 2 or 3 day training seminars. A great way to advance your knowledge and help maximize the use of the instrument, gain from the benefit of of all this experience that we have in order to create ideas for how you might want to approach your challenges. We have a very nice rental program. The instruments are more difficult to demonstrate on site prior to the sale because it takes a while for the instruments to get put up on the bench and equilibrated, and then the experiments can be 24, 48, 72 and more hours. So It's not an easy instrument to bring into the lab, set up on the desk or on the lab bench, and a couple hours later have data. It just doesn't work that way. So the rental program is nice because it allows you to put an instrument into a laboratory environment in your application with your folks and your sample prep capabilities, and use an instrument for 2 or 3 months and assess an application for a very nominal monthly fee where 100% of the principal is applicable to the purchase. So a very nice way to assess an instrument for an application. And secondarily, it can be used if you've got instruments now and you've got a big project coming down the road and you need additional capacity. It's a great way to add some additional capacity for 2 or 3 or 6 months in order to run a specific project. It's also done directly through Calmetrix, not through a third-party leasing company, which provides great flexibility as well. And finally, we can do sample analysis. It's very context specific, and it's something that we would need to talk to you about. Some samples just can't be shipped. There's sample prep issues. So there are situations where we can do it, but we really would need to speak with you individually to decide whether it's appropriate or not. So with that, I'll close. This is my contact information. I'm happy to answer any questions related to how the instrument can be used in this application area. I can answer questions for you in terms of the instrument itself or the rental program or running samples and so forth. If it's not me, then I can put you in contact with the right people. So here's my contact information. And with that, I will close and thank you. I hope that you found this webinar informative and now have a better understanding of the power of this this technique of isothermal calorimetry and how it can be applied to food safety and spoilage. Thanks.